Thank you. I'd like to welcome Omer and you all to the live question and answer session of the seminar. Please do ask your questions in the box on the screen and we'll ask the most popular questions. So if there's one you particularly like to see answered, then please use the upvoting function. Thank you so much for your talk, Omer. I'd like to start the questions because I was particularly interested in the plastic cell states in glioblastoma. And how do you actually define what a cell type is? And then how far do you keep splitting these cell types into further subtypes and then further subtypes again?
Thanks so much, Julia. I think it's a fantastic question. And I think we probably don't have a very satisfying answer to it. I think, this is, I think, sort of an existential issue that the field of single cell biology deals with. Like, how do you, take single cell level transcriptomic data sets and draw the boundaries between cell types and decide, which gene expression programs or identities are biologically relevant or not. I think, and this is sort of, I think this challenge is codified as sort of as splitters versus lumpers. And you see it, I think, especially in the brain where there's such a huge multitude of different neuronal and glial cell types. I think one way, one sort of potential solution to defining what cell types are and, identifying them in more sort of challenging situations like GBM, where our prior biological knowledge of what relevant cell types would be is poor, is to think about defining cell types based on multiple streams of information. So not just simply relying on their transcriptome or their epigenome, their sort of somatic mutation landscape, or their tissue microenvironment, but actually putting all these different types of information together using, new computational approaches. And I think, in the case of GBM, it would be very interested in sort of thinking about questions like, if we could really identify these, transcriptional cell states from the single cell data, do these actually associate with specific recurring tissue microenvironments across the tumor and across patients? And I think that type of multimodal approach to defining cell types and cell states will be quite future-proof and rewarding.
That's brilliant, thank you. I think that there are similar themed questions coming up, so it's really useful context to have. Thank you. I'll pass you to Rosa now.
Thank you, Omer. Building on what you were saying, I was wondering if you can tell us a bit more about the challenges on data integration, for example, integrating different samples or different experiments.
Yeah, absolutely. Thank you, Rosa. It's I think the challenges are numerous, right? So if you were to start by, I think if you were to sort of start by integrating just one modality, right? I think a lot of the challenges that were faced and dealt with by the single cell RNA-seq community will apply to many other modalities. So, thinking about batch effects across, different experimental runs, thinking about, differences between, different donors, thinking about the challenges of working with really large data sets of lots of cells. I think all of these will apply to spatial genomics eventually, right? As you think about, putting together information from tissue sections from multiple donors, tissue sections, numerous tissue sections representing different parts of an organ, and large data sets that consist of hundreds, thousands of tissue sections with hundreds of thousands and millions of tissue locations. Regarding, I think, multimodal data integration, I think a few of the things that we've noticed, and I'm specifically, if I specifically talk about single cell and spatial transcriptomics, a few of the things we've noticed that make a real difference are recognizing that, single cell and spatial transcriptomic measurements are not quite the same thing. So beyond the fact that you don't have the same resolution and you're not doing the same thing, the techniques you're using to sample transcripts are almost fundamentally different. So the sensitivities of the assays, the sort of like the background noise in the assay, these are all different things. And we've found that with self-allocation and other tools, they're most successful when you actually account for these technical differences successfully. What I kind of think about moving forward is that, sort of following up on the first question too, thinking about truly multimodal definitions of cell types. I think what will be most challenging, but again, most rewarding perhaps, is thinking about, integration protocols that don't box in each of these modalities, but take a truly sort of interactive or iterative approach in combining information from single cell transcriptomics, spatial genomics, and say epigenomics, and use all the streams together simultaneously rather than try to relate annotations from each category.
Yeah, I wonder if maybe we can follow up with another question that is quite similar. And in this particular case is for the glioblastoma project, and it's more a specific question on integration, and which is your spatial whole genome sequencing readout, and how you specifically are going to integrate this with RNA and ATAC data.
Mm-hmm. We're quite excited about that. So our spatial whole genome sequencing readout is not our product. So it's a wonderful pipeline developed by the CHASM program at Sanger. It's basically a low input. It's basically a very sort of precise laser capture microscopy pipeline coupled to low input whole genome sequencing. And the way it works is a bit of a Swiss Army knife of spatial DNA profiling. So often you would start with a piece of tissue, you'll do a histological stain, and you would sort of cut out with LCM a histological structure that you deem as relevant to the biology of the healthy or the disease organ. In our case, what we are thinking about is sort of two sort of pronged strategy to try to integrate that with transcriptomics and with epigenomics. So first off, we'll probably use LCM as a kind of a coarse spatial resolution version of Visium. So instead of taking a tissue section and cutting out histological features, we will actually sample a grid from each tissue section, right? So that will, we will apply that to tissue sections whose sort of consecutive tissue sections will be sampled with Visium. So that would give us a better sort of one-to-one correspondence with the nature of the data modalities. Second, we're kind of thinking about we have formulated, I think, our sort of strategy to analyze somatic mutations in the case of ATAC data, perhaps a bit more. And I'm hoping that this would be applicable to, LCM WGS down the line. With somatic mutations, what we're really hoping to do is actually to harness the fact that when you're performing single nuclei joint ATAC and RNA sequencing, you're actually capturing a physical portion of the genome from each single nuclei. So we're actually going to attempt calling mutations in transcription factor binding sites as well as transcription factors themselves using that. And I think with those types of approaches, if you are sampling, right, both the genome information and the transcriptome information in the same cell, then the integration becomes much easier.
Yeah, thank you very much.
Great. Omer, I mean, it's somewhat related to the GBM project, but I think taking maybe a broader view. So the question is, does the location differ, I believe, of cell types between samples from healthy and diseased patients? And do we see the same segregation of cell types? So I guess the question is, can you frame that in, what do we know about various diseases, cancer and other diseases, in terms of how much variation we saw in location or cell types?
I think very much so. I think, location of cell types is synonymous with more or less the anatomy and like morphology of tissues, right? And I think these have traditionally been out of the reach of genomics, but I think biology of organs or tissues is this deep entrenched view that, right, like solid organs have cells that are assembled into very specific and particular tissue microenvironments. I think there are great examples across different diseases on how location matters as well, right? So if you were to sort of, for example, talk about COVID, I think, which I presented a little bit from, I think there's this now sort of you're starting to build a spatially resolved understanding of how the immune damage occurs in the lungs. And you sort of, if you were to sort of piece together the evidence we have from postmortem cases, we are starting to see that, we're starting to sort of speculate. We can now, I think, safely speculate that, the damage starts at the blood vessels that are, surrounding the alveolar units. And from there, it sort of, starts to spread out and sort of triggers the cycle of inflammation. And I think, for diseases like cancer, I think I like to think that some cancers have had sort of appreciated tumor microenvironment for a while. Others haven't. I think GBM probably falls into that second category. One of the reasons we're excited about it. As I was telling you guys before, I think if you were to look into some, GBM tumor types, you actually see that the cell types, the most abundant cell types present in them, are actually not tumor or malignant cells. They're actually sort of the microglial, innate immune cells of the brain infiltrating them. And GBM is probably also another good example on how, say, different parts of the tumor, different spatial environments show different characteristics. In fact, even before single cell or spatial transcriptomics, we actually knew that different parts of a patient's tumor could have completely different characteristics of GBM. They would be more or less diagnosed. If you had that resolution, you could actually diagnose a patient with multiple subtypes of glioblastoma just by looking at different parts. So I think, yeah, absolutely, both in health and disease, tissue microenvironments and spatial structure are huge.
Great, thank you.
Thanks, Omer. I've got a question now about, are there advantages of using spatial transcriptomics to study cell-cell communication? So can you observe ligand and receptor interactions in your data, or is that pushing the technology too far?
No, I think absolutely. I think looking at cell-cell interactions with spatial transcriptomics is one of the great promises of the technology, and it's something that we're doing right now. So I think people like Roser had shown a while ago now that you could actually look at that information in single-cell genomics data. And as part of a wonderful collaboration we've had with Roser, for example, we could actually sort of integrate spatial information into that type of analysis. And in the human endometrium, we could sort of we could integrate single cell genomics with spatial transcriptomics to put the cell types where they're supposed to be within the tissue, identify which cells are more likely to neighbor each other, and actually predict cell-cell interactions in a much more sort of accurate, cognizant, spatially cognizant manner. And I think it's no with spatial transcriptomics and predicting cell-cell communication, there are probably a couple subtleties. I think to date, the most successful approach has been what I've just described that we did with Roser's lab, where you do a lot of your cell-cell interaction analyses with a lot of your single-cell data, but you actually map your single-cell data into space to prioritize which cell-cell interactions are likely happening. Different spatial methods have different sensitivities to actually detect the expression of cell-cell communication molecules. So that's like one concern. I think, again, if I go back to a bit of a broken record about this these days, but I think, again, multimodal observations where you don't just have spatial transcriptomic data or single cell data, but you actually have them both of the same tissue at the same time are, again, going to be the most powerful here.
Well, that's really helpful. And it relates to another question that we've got here, is whether you can start to build 3D models of tissues if it's possible to use serial sections and all that multimodal data to build a 3D picture of what's happening and the cell-cell interactions and where they are.
Absolutely. I mean, I'm a keen believer in that. I think, you know, at the moment, right, you can use sort of lower dimensional, sort of lower plexed methods to already take, you know, sort of three-dimensional tissue samples that span, you know, dozens of cell layers, and, you know, optically clear them and image them through, etc. I think, you know, doing things like serial Visium sections, you know, intercalated with, you know, tissue sections that are profiled with single cell and single nuclei transcriptomics are protocols that we're already using, you know, in projects. And I think it'll be really interesting and awesome to really think about, can we now scale this to an entire human organ? And I think that's where maybe some of the challenges start to come in. I think the proof of principle for a lot of these types of approaches is there, but you know, could you actually, for example, map an entire human brain, which is smaller than what, you know, most people would tend to think. It's certainly, you know, the human brain in terms of its dimensions is smaller than I thought it was. I'm not just talking about mine. But, you know, you're still talking about, you know, an organ that is about 20 centimeters deep, 17 centimeters wide. So, you know, how many Visium chips is that? How many nuclei is that, right?
Yeah. Well, that's a super answer. Thank you.
So there is another interesting question about the subcellular structures using spatial transcriptomics, and about what are the practical and theoretical limits to go into that. Also thinking about future directions of spatial transcriptomics technologies.
That's fantastic. Yeah, I mean, I think subcellular resolution is definitely within the reach of the technologies. So I think, you know, I didn't get to talk almost at all about the imaging type of work that we do at my seminar, but, you know, I think broadly spatial transcriptomics methods come in two flavors. They're either sort of, you know, positional RNA capture from tissues and spatial RNA-seq based, or they're imaging based, where you tag, you know, RNA or protein molecules, and, you know, you can do iterative rounds of labeling and imaging to really measure, you know, dozens of different transcripts, or, you know, dozens, hundreds, thousands sometimes transcripts. I think with imaging, obviously, you know, you control the resolution that you image the samples. So there are many examples of high subcellular resolution, you know, submicron resolution image data sets. Has there been, you know, I think really sort of paradigm shifting work that's been done with that. I think we're just really starting to think what to do with that type of information. You know, I think we don't necessarily have, I think at the moment, the best sort of both sort of statistical pipelines, the best statistical pipelines to deal, to measure the subcellular localization of hundreds and thousands of transcripts and millions of cells and make sense of that in the context of a very large tissue. What's been very surprising to me, and it's really amazing, has been actually the sort of increase in the resolution of sequencing-based methods to make a claim on looking at subcellular co-localization. So I think, you know, with the current Visium assay, obviously with a 55 micron resolution, unless you're looking at truly massive cells, you're not going to get any type of that. You're not going to get close to that structural resolution. But now we're actually looking at a very exciting market next year, where both the upgrade to the Visium assay, the HT assay with the 4 micron resolution, but also now some decent competition. For example, the BGI nanobolcy, which gives like a whopping, I think, 200-300 nanometer resolution, will be there. And I think they will actually put us in now the ballpark of answering some of these subcellular localization questions at an unbiased transcriptomide level. I think the sensitivity of the methods will still fall into play. Maybe that's where imaging-based technologies will have an advantage still for several years. But yeah.
So we need to start thinking about all these subcellular resolution questions for our tissues.
Yeah, I think a part of the problem is that, sometimes I feel like we can barely analyze single cell level data. So when you start thinking about, or, we can barely understand how single cells come together to make tissues, right? So when you start now going like, okay, now I'm actually going to go within each cell and I'm going to see I'm going to see if the apical or basal localization of the RNAs in these millions of cells across this tissue environment have a significance. It's a challenging question.
Thank you so much, Omer. We would really like to thank you once again for your presentation and joining us for the Q&A session, and also to the audience who joined this virtual session. So we have seminars lined up for November and December this year. So next month, we will welcome Trevor Lolly to talk about the human microbiome. So please save the date, which is 17th of November. And also a reminder that these seminars are archived and that on this webpage for future viewing. So we have delighted that this series has allowed us to share Sanger science with a diverse scientific audience around the world. So thank you for joining, and we look forward to seeing you in November.